Can you hear me? Everybody hear me?
Yep, you sound great.
Yeah. Okay, there's an echo. Somebody's getting an echo, but okay. Well, thanks for inviting me. um, a fair amount of my job is to collaborate with biological and agricultural researchers, and so I've spent a fair amount of time over the last umpteen years working with plant breeders and professors and students. And so maybe I have something to offer, although I mean it's going to be pretty obvious that I'm not a plant breeder or a geneticist. So please bear with me. Uh, the basic objective of this webinar will be to review a little of background in terms of the statistical side of field design, and then hopefully talk about some concepts and some designs that would be useful in your work as a plant breeder. Um, so in my simplistic way of thinking about plant breeding, of course, you people do lots of different things, but it seems to me there's two major things that plant breeders do is to, first of all, create and describe and identify variability. Now, what I mean by that is, of course, you create genetic variability by making crosses, and then you want to describe it and identify it. Is it really genetic variability that you're looking at, or is it environmental, or is it the interaction between genotype and environment? And then maybe the most important thing for a plant breeder is to be able to select superior genotypes. And so the whole point of all these field designs is to provide data that will be useful to farmers down the road. But if you're going to produce things that are going to be useful to farmers, you're going to have to evaluate them in farmers' own environments. And what does that mean? You're going to have to do field trials of one kind or another. And one of the problems about field trials, of course, you all know, is that there's huge amounts of field variation. For lots of different reasons. And that's just within a field. And then if you're doing multi-location trials, which a lot of you people are doing too, then you got all that environmental variation comes. There's huge amounts of variation. And the reality is that you probably have lots of entries to evaluate, especially if you're early along in the breeding program. You maybe have hundreds, maybe even thousands of entries you want to evaluate. And the reality also is that you want to look at differences between the entries, but most of these differences are going to be very small. Uh, Probably less than a bushel an acre, probably substantially less than a bushel an acre. So there's two bad things going for us here. You got huge amounts of variability and you want to pick up small differences. So whatever we do, you need to do it smartly. Let's go back and review a little bit the concepts of field design and, well, statistical side of it really. Is if you think of the major objectives of any kind of experimental design, what you want to produce are unbiased and precise estimates of the entry differences or the treatment differences. What you want to do is see if one is different from the other. Okay, preferably one of them has higher yield and you choose that one. But you'd also like to have a design that's cost effective. You don't want any bigger than it needs to be. These are huge experiments as they are. You don't want any bigger than what they are already, and you'd like to make them smaller if you possibly could. And not only that, just to explain the ideas to other people, you need to have these very simple. Agricultural experiments can be the most complex things known to the human mind, I believe. And so you need to keep them as simple as possible. The reason we can have such complex designs in agriculture is because you can have so many factors. In, say, clinical trials and human studies, you don't have very many factors usually. Lots of other areas, you don't have very many. But boy, in agriculture, you really can. So the essential characteristics from a statistical point of view is that you better replicate each entry. You better have several plots per entry. And in order to get an unbiased estimate of the differences between the treatments or the difference between the entries, and an unbiased estimate of the error variance, you better randomize. And I'm assuming most people here know about replication and randomization. And then finally, because you have lots of field variation, um, blocking is an important issue. If we got lots of variation in the fields, then we need to break the field up into smaller areas that are called blocks that are hopefully more uniform within the block. And then you can apply all your entries or plant your entries within a smaller block and hopefully have a more precise estimate of what the differences are. And so I'm going to make some assumptions here before I go on. First of all, that you have many, many entries. And some of the examples I'll give, there are not that many entries, but the point of this is just to give you an idea. Um, And assuming that all the entries are equally important. We don't have any that are more important than others. Okay, so we want to be fair to comparing all the different entries. And further, I'm going to assume that a complete block design is inefficient. If you have a lot of entries, so for example, if you have 144 entries, a block size of 144 maize plots is just too big. I mean, you know, maize corn plot's pretty large, and so if you put 144 plots together, that's just too big of a piece of ground for practically to be a very uniform block. So complete block designs, or randomized complete block designs, are inefficient. And so you need to use block sizes that are quite a bit smaller than the number of entries you have to evaluate. Okay, so we'll first talk about incomplete block designs. Maybe I should stop here to see if there are any questions, or you can hear what I'm saying, or can't hear what I'm saying, or is okay? Yeah?
I think so.
Nobody's really complaining, I guess, at this point. Okay.
There's
Looks
one
like there's a question.
just confirming that when you say entry, you mean the line.
It could be, yeah, when I say entry, it could be the line. It could be a cultivar. It could be a family. You know, it could be, you know, any character or group of individuals that you group in a way that's relevant to you. Okay? Excuse me. Okay, other questions? Okay, so incomplete block designs. Well, the most important thing about incomplete block design is you want to choose the design, in other words, what entries are in what blocks, to minimize and to equalize the variance of a treatment difference. Okay, we want unbiased estimates of the treatment differences, but we also want to minimize and equalize the variance of the treatment differences. And so good incomplete block designs will do this both. If you're blocking in the correct way and you have a good incomplete block design, you'll minimize the variance, and with certain types of incomplete block designs, you'll have equal variance for all those different entry pairs. So incomplete block designs are also good in the fact that you can increase precision with the same number of plots as you'd have with a randomized complete block design, or in fact, you can decrease the cost with the same power. Okay, in other words, you can have the same probability of showing differences between entries, but probably get by with less area, less plots, less money. Okay, so incomplete block designs are useful things to think about when you talk about field experiments. First of all, let's talk about balanced incomplete block designs. It's possible to show that if you have a balanced incomplete block design, that is, every entry pair appears together in a block equally often, what that does is that it equalizes the variance of all the entry differences. And so, and also if you're blocking in the correct way, then you minimize the variance. So an example, a simplest possible example almost of the balanced incomplete block design is the plan here that I have. Now see if I can click on my my mouse. How do I click my mouse on? Draw?
Nope. Right there.
Okay.
And then you have to click.
Okay, so here's the plan. Okay, let's assume that I only have 4 treatments or 4 entries called A, B, C, and D. And they have blocks of size 2. So here are the blocks. Here's the first block with A and B in it. Here's the second block with A and C in it, and so on. Okay, well, if you look at how the entries are arranged in each block, I have A is with B in one block, A is with C in another block, A is with D is in another block. Okay, so A is with every other treatment one time, okay, together in a block. And it's the same for all pairs of treatments. Okay, the analysis of variance is also very nice and easy to understand. You just in this example, you have 6 blocks. You have 5 degrees of freedom per block and 3 degrees of freedom per entry, and error of 3 degrees of freedom. Probably not enough degrees of freedom to really have much power, but it's a valid analysis. Okay, so this is the simple idea of a balanced incomplete block. We want all pairs of entries to occur equally often with other entries throughout the design somewhere. Okay, well, if you have more treatments, then you need to be a little bit more sophisticated. And an example is a balanced lattice. Now, lattice is where the number of treatments, in this example 9, is equal to the cube of the block size. Block size here is 3. And what I mean by block is I have 3 plots in a block. And in this first block, I'd have entry 1, 2, and 3. So in each replicate, So in the first set of 3 blocks, this is a replicate, and I'd have all treatments in that first replicate. The second replicate, I'd have all treatments in this together and so on. So I have a total of 4 replicates here, which would be a total of 36 plots. Okay, well, this is a balanced incomplete block design. If you notice with treatment 1, it's with entries 2 and 3, 4 and 7, 5 and 9, and 6 and 8 together in the same block, exactly one time. Okay, so that's a nice design. It minimizes the variance if you're blocking the right way. It makes the variance between treatment differences all equal. It has a very simple analysis, and here it is down here. Another thing that's nice is if you find out the incomplete blocks are not very useful, you can maybe remove the blocking within rep effect and just analyze it as a complete block design. This is what's called resolvable incomplete block design. The idea is resolvable as all the treatments appear one time in a replicate. Okay, and they're easy to work with, easy to manage. Not only that, if you lose a replicate, say if you're harvesting and you don't have enough time to harvest all the blocks, well, maybe you can just harvest the first 2, and you can still get some valid data out of this. Okay, as long as you do it on a block-by-block basis. So resolvable entries are sorted into complete replicates, and the advantage are the analysis is easy and it's easy to manage and so on. But there's a big problem with balanced incomplete block designs. They take too many blocks. In other words, this idea of balance, of getting equal occurrence of all pairs of treatments within a block together, it takes too many blocks, especially if you got a lot of entries you want to evaluate. So the simple solution is discard some replicates. So if you think about the previous slide where I had 4 replicates of 9 entries in each replicate and 3 block size of 3, we can just throw out the last 2 replicates. And this is a valid incomplete block design. However, it's not balanced. Notice that treatment 1, entry 1, appears with entries 2 and 3 in the first block on the left. And the next block and the next replicate, entry 1 occurs with 4 and 7. It never appears with 5 or 9 or some of the other ones. Okay, so it's not a balanced incomplete block design. And so that means that we're going to have more precision about comparing some pairs of treatments than we do with others. So we're being unfair to some of them. But you know, that's life. We don't have enough plots a lot of times to run a balanced incomplete block design. This is the idea of lattices. Yeah.
Unclick the arrow to make it go away.
Okay, so lattices. I'll just leave my arrow there and click on it as I use it. I hope that's okay, everybody. So the lattices were the first field designs that were developed by Frank Yates, probably back in the 1930s or something like that. And the one I just talked about is just called a square lattice, where the number of treatments is a square of the block size. K is the block size. And so if I have a block size of 3, then I have 9 treatments. Now we can also have things called a cubic lattice. A cubic lattice is where the number of treatments is a cube of the block size. So I could use a cubic lattice, and I won't go into those here. You can look in Cochran and Cox's book on cubic lattices. You can also get a quartic lattice, where you have the number of treatments is raised to the 4th power of the block size. Anyway, it's possible with these lattice designs to generate, to evaluate an enormous number of entries. Notice, so if I have K is equal to 10, block size of 10, I could evaluate 1,000 entries in an incomplete block size 10, but it's going to take a lot of space, but you certainly can do this. But one of the problems with lattice designs is they're just too restrictive, okay? And so that's given rise to field designs that people have used over the years called alpha lattices, or just alpha designs. Okay, and they were introduced by Patterson and William in '76, and I think as Emlyn Williams' PhD dissertation is where these came out of Edinburgh. And the idea about alpha lattice is the number of entries and number of treatments T is a multiple of block size. Say, for example, if I have block size of 4, and if I have 24 treatments, that is 24 is equal to 6 times 4, 4 being the block size, then I can generate an alpha lattice. So they're not nearly as restrictive. So down here, if I say an alpha lattice, they're not nearly as restrictive as what we had with the lattice designs that I have up here. So they're much more flexible. We can evaluate many more situations of given block size and number of treatments. Okay, and we'll give you an example of what a alpha lattice looks like. So an example of 24 treatments, block size of 4. So I have the first, here's the first block. I have entries 1, 7, 13, and 19 in replicate 1 and so on. Okay. And so we can generate incomplete block designs with alpha designs or alpha lattices. And so these are nice, but the major problem with these is they're not balanced. Well, that's life. You probably don't have enough money to get balance anyway. So if you have a lot of entries and block size is fairly small because you want to remove a lot of the variability in the field, it's probably a good idea to think about an alpha lattice if you can do it. Alpha lattices are very flexible. You can use them with lots of different numbers of treatments and block size. One thing that's nice about alpha lattices is that they can be used when the number of treatments is not necessarily a multiple block size. I'll give you an example here where you have 21 treatments where block sizes of both 4 and 3. Okay, so you'll notice, say, the first 3 blocks here in rep 1 all have block size of 4. But the last 3 blocks in rep 1 all have block size of 3, and it's easy to generate that design by just getting an alpha lattice with 24 treatments and eliminating the last 3 treatments. So they're easy to use, and they're very flexible and fairly easy to understand. You can generate them with software. The one I'm most familiar with is probably AlphaGen or Alpha Plus. I've also used Gendex. That's easy. In fact, I don't know if the TCAP people have access to Gendex. I know Ibrahim has access to it.
We
And
use
so-
Agribase.
But there's others too, yeah, Agribase, and I think you can do some of these with SAS, and there's one called Sikta Design, which is the one Emlyn Williams uses, and although these are not free. So, and also probably R has the ability to generate alpha lattices. I didn't put that down there. R is free, and so those that if you know about R, you could use R to generate alpha lattices. Okay, and so the problem with the alpha lattice, I said before, is you can never achieve balance. So really, practically speaking, if you have a large number of entries, it's probably okay. So I wouldn't ever use an alpha lattice if you have a smaller number of treatments, because ideally you'd want to use a balanced incomplete block design or a balanced lattice if you could, and you can never achieve that with an alpha lattice. Okay, I'm looking at some questions here. Who might have the R code for generation of the alpha lattice? You can, I think there's a package called Agro, it's not Agrobase, but it's Agro something. I'd have to look it up. I think what you could do is just go and Google R code and alpha design, and it should come right up. And there's a nice package that they can generate lots of different kinds of designs, and it's all free. And I haven't used it that much, but hopefully the problem with the R code is, of course, has it really ever been validated? We don't know. So you need to take it upon yourself to make sure it looks reasonable. Maybe we'll stop at this point. Are there any questions? Other questions? Okay. So those are alpha lattices. Now, sometimes, wait a minute here. Sometimes that you have more variation in the field than what you really know how to deal with. It could be you have variation in 2 directions. I've just been talking about blocking in 1 direction at this point, but it could be that you have variation in 2 directions. You could have a moisture gradient in 1 direction, and north-south, and a fertility gradient in the east-west direction. And gosh, how could you take care of that? Well, we could use Latin square types of designs, and that's what row column designs are. They're kind of a generalization of a Latin square. Here's an example of a Latin square. We're blocking in 2 directions. And so each row is considered to be, in this case, a full block. Each row is a full block, and each column is a full block. That you notice in each column you have all 4 treatments, and each row you have all 4 treatments. And in a sense, this is a perfect design. It's kind of like these lattice designs. They're perfect in a mathematical sense. The problem is, and here's the analysis of variance, so the analysis of variance is very easy to use. The problem is that many times you don't have the right number of rows and columns, you don't have the right number of entries, but you do want to try to remove variation in 2 dimensions. You'd like to remove variation, say, in the north-south direction and the east-west direction. So it's useful to try to use some of these ideas. So there's some defining principles in row column designs that we can use. And ideally, we want them to be Latin squares. Well, a Latin square means that each entry appears an equal number of times in each row and each column. That's called orthogonality in a mathematical sense, or perpendicular in a sense. And so that's what we had in the first slide, or first slide with row column designs, is a Latin square is orthogonal in both directions, in both the rows and the columns. But if you can't get that, then you'd like to get all the treatments, say, in a row, appearing in a row, and then a balanced incomplete block design maybe in the columns, or vice versa. That's the second best thing, if you can get that. And if you can't even get that, then you'd like to have the joint occurrences of each pair of entries in a row or columns an equal number of times if possible. So let me give you some examples of these. So a cyclic row column design, if you're interested in cyclic designs, is a very nice book by Williams and John and Williams on cyclic designs. And that's where this one comes from. A cyclic design, let me explain a little bit what this is. This first block, incomplete block, is what's called a generator. And notice that as you go down the row, each row will successively have a larger number. And so the first block is called a generator block. And so we have one generator block in rep 1 and another generator block in rep 2, and then we just cycle the numbers down. So cyclic designs are fairly easy to generate, and in certain numbers of them, they generate nice row column designs. If I want to block in 2 dimensions, look in the columns. All treatments appear in the first column, and in the second column, and in the third column. Okay, so we have orthogonality in the columns, and in the rows, we have, turns out to be, a balanced incomplete block design. The first person to generate these designs is a guy named Yowden, working with tobacco plants back in the 1940s, I think. So cyclic designs can be used for row column designs, and this kind of gives you the idea of what I mean by trying to get orthogonality, say, in a column, and a balanced incomplete block design in the rows. Well, a lattice square is an example of the third case, whereas you can't get orthogonality either rows or columns. But what you want to do is you'd like to have each treatment appear with each other treatment someplace in either a row or a column, if possible. It's kind of this idea of equal joint occurrence. And so we can generate a lattice square by starting with a balanced lattice. And notice that if I take the first row, the balanced lattice, I get treatments 1, 2, 3, 4, 5, 6, 7, 8, 9 in the first 3 blocks. Okay, but let's look at the columns. What do I get in the columns? If I consider a column to be a block also, notice that the first column in this lower, after I arrange the rows, the first column has 1, 4, and 7 in the first column, which is exactly the same as the first row in the second rep. And notice that in the second column of the first rep of the balanced lattice, it's 2, 5, and 8, 2, 5, and 8, the same exact treatments that I have in the row in a balanced lattice. So using a balanced lattice, I can generate a lattice square. And it has this very nice property of, notice that treatment 1 is with treatment 2 and 3, 9 and 5 in a row, and it's also with 4 and 7 and 6 and 8 in the column. That means treatment 1 is balanced either in a row or column somewhere in the design. Okay, well, that's not always possible to get, but this is a nice design if you can achieve that. But row column designs are kind of a generalization of these ideas. So if you have variation in 2 directions and you want to try to remove it if you can, using row column designs would be an excellent choice. There's other ways that you can take alpha lattice and just generate a regular alpha lattice and try to get as close to a row column design as you can. And this is what's called being a Latinized design. Here we're trying to balance out the number of entries in a row, and notice that 1 is only with 7 one time in any particular row, okay, or any particular column. 1 is never with, well, unfortunately, it's with 6 in the same column here one time, but there's nothing we can do about it. So those are Latinized alpha lattices. Also, Williams showed how, in '86, showed how to, if you have contiguous plots that look like this, say you got rep 1, rep 2, and rep 3, and then so we have long rows of plots that look like this, we can block in 2 directions by using the ideas of alpha lattices. Okay, are there any questions? All right. Now, what I've talked about up to this point is the idea that we need to replicate each entry in one way or another. And the truth of the matter is you may not have enough seed to replicate all these entries. Okay, not only that, you may not have enough space. You've got 1,200 entries you want to evaluate. My gosh, you don't have enough money. You have to evaluate them all. But are there other ways of not having to replicate every entry? Okay, and this gives the idea of screening designs. In fact, the expected genetic gains, making some assumptions, can be increased by screening more entries and not having as many plots devoted to replication. And so the idea is you can only replicate a few checks, if at all. And to do this, there are several different types of designs that people use, such as augmented designs and unreplicated designs. Okay, so let's talk about an augmented design. These were first proposed by Feder, back in the 1950s. And the idea is you have some check entries, okay? And we replicate the check entries in a randomized complete block. And then we have experimental entries where we only have one plot of each. And we augment the experimental entries with the randomized complete block design that we're using for the checks. So say, for example, I have 3 checks, A, B, and C. And what I'm going to do is I'm going to put A, B, and C in a randomized complete block design across the top of this scheme that I show here. And let's assume that I have 36 other entries. Okay, well that means if I have block sizes of 12 altogether, okay, I can evaluate 9 unreplicated entries in each block, which allows me to evaluate 36 unreplicated entries by replicating the checks. And what this does, it allows me to get a valid estimate of error variance and test differences between the entries, even though they're not replicated. We can do this also, we could use an augmented incomplete block design. Instead of having complete blocks, we could put the checks as an incomplete block design. Remember the first balanced incomplete block design we talked about looked like this. Okay, so we could say I've got 4 checks, I'm going to put those in blocks of 2. Okay, I could use these as the repeated or the checks and then augment with more entries. So I could do this with 60 entries and six blocks. So I could evaluate 60 experimental entries with just 4 checks, and this is an easy design to use. Now notice you're only blocking in one way. Okay, we only have one block, so that each block in this example is 12 plots. But this is a fairly easy design to use, and people have used a lot over the years. We can also apply these ideas of row column ideas to augmented design. So we could set up not just as randomized complete block design, but notice I could set up a Latin square on the checks, okay, and then have plots of size 11, and then just augment each block of size 11, and augment each block with the experimentals 1 through 10. And so in this example, I could evaluate 160 unreplicated experimentals with 4 checks in a Latin square with 11 plots in a block. So I'd have a total of 16 blocks altogether. I can also do this with an augmented lattice square. And so we can balance it basically in 2 directions in a sense. What we could try to do is set up the checks in such a way. So in this example, assume I have 8 checks. I could set up in such a way as I could remove the row-to-row variation within a replicate, and I could remove the column-to-column variation within a replicate. Okay, so I can use this as a Latin square type of a thing where you can remove both row and column variation with an augmented design. So these could be very useful too, especially if you need to block in 2 directions and you need to evaluate a lot of experimental lines and you don't have enough seed to replicate them, or something similar to that. Williams went further and said, oh, we could use an alpha-alpha lattice design. Okay, so and he gives an example here. We have 24 checks and 120 experimentals. We can still actually use a row column design in an augmented sense. Okay, so you can see his paper if you're interested in looking at these kinds of things. Maybe it'd be worthwhile to talk about what the ANOVA looks like for these. You very quickly, if you're talking about an augmented row column design, you very quickly run out of degrees of freedom to account for the row and column effects. So what Feder suggested to do is just to fit linear row effect and linear column effect and fit interactions. And so that would be the 3, you'd have, if I have 3 replicates, I'd have a degree of freedom for linear for each of the 3 replicates, a degree of freedom for row, and do freedom for linear for column for each of 3 replicates, and then 3 by 3 is 9 degrees of freedom in terms of how those things interact, and hopefully that removes most of the row and column effects. And then we can evaluate the entries with the remaining ANOVA with 31 degrees of freedom for error. So that's an easy ANOVA to use, easy to understand, and it gives enough degrees of freedom to have a reasonable test. There's some newer designs that people have proposed and been using recently that I've noticed is they're called PREP designs. And as far as I can see, there's a paper by Smith and another paper by Cullis, a couple people out of Australia. Some of you might know them. And they were, I think, the first ones to really talk about these designs, although my guess is that people have used these things over the years and just haven't formally talked about them. Ideally, the idea is that you don't want to have any checks at all in your experiment. You have virtually no checks, but you do have enough seed where you can replicate some of the entries. And so what you do is in maybe each site or each replicate, you only replicate, say, a certain proportion of the test entries. Okay, and these are really quite flexible, so you can use them with any number of entries. They can be used in place of unreplicated designs, which I'll talk about here in a minute. And they also lend themselves to multi-location experiments, where you can replicate different entries in each location. So here's an example of a PREP design, very simple one. Say we have 40 test entries, and what we want to do is we want to replicate 25% of the test entries, which means I'm going to replicate 10 entries. In this first site, I'm going to replicate each of the first 10 entries. I'm going to replicate them twice. Notice here entry 1 appears first in block 1. Now here the column is the block. The column is the block, and entry 1 appears once in the first block and appears once in the last block. Okay, and so I can replicate each of the 10 entries, replicate those twice. And it looks very similar to just an augmented design, except what I will, and then I'll put the remaining unreplicated entries within each block and just balance them out the best I can. Okay, it's very similar to an augmented design, except what you do is you replicate different entries in each site. So say, for example, in site 2, we replicate entries 11 through 20. In site 3, you replicate entries of 21 to 30 and so on. So in a sense, they lend themselves to multi-location experimentation. You could do the same thing with a single site too. I mean, you could just have full replicates and do the same thing. Instead of calling these sites, you would call them replicates.
Do you have a question there?
Is there an optimum value for P? Well, that's a good question. I don't know. I mean, you can read the paper by Cullis. He does talk about this, but he uses some specific assumptions about the nature of the variation in the field. And he does talk about that a little bit. I think that's a good question. Personally, I think this is an area for research that we need to look into more. And I frankly think that we could get some good dissertations on answering some of these questions with PREP design, because they seem to be so popular at this point. So I have to say, Scott, I'm sorry, I don't know. I can't answer your question. Are there other questions? Okay. Okay, so a PREP design could also be used with alpha lattices. And Williams suggested using alpha designs to generate a PREP design. Okay, and basically what he does is uses the generating array to generate a little alpha design for the checks in each site. So if I have 10 checks in site 1, then he uses an alpha array to generate how the blocks or how the entries go into the blocks for the first 5, and then he just puts these things together with the other 10. Okay, and he puts them together. Anyway, you can read in the paper about this, but you can notice that we replicated the entries 1 through 10 in site 1 and replicated the entries 11 through 20 in site 2, and we can use an alpha design to do this. So if you've got lots and lots of treatments, lots and lots of entries, you probably need some more systematic way of doing this, and just kind of instead of doing it off kind of the top of the head what I'm talking about here with the first design. It's probably worthwhile to look at using alpha PREP designs to generate these kinds of things. Not only that, you can Latinize these alpha P-rep designs. If you think there's variation in two directions, okay, in your field, then you certainly could get an alpha P-rep design and then Latinize it. And in this example, I show how you can maybe switch the original design to just switch these rows, and so you don't have an entry appearing in a row any more than once. Okay, so we could probably Latinize these things too to get to remove some of the variation in both directions. Also, Colas talks about, when he first introduced these ideas, he talked about a special P-rep design, and what he did is he used resolvable in the replicating checks, and he replicated each 15 checks a couple times, and he pre-specified spatial correlation in both directions with an AR1 in one way and AR1 in the other way. The AR1 means an autoregressive one. It's a standard time series type of model. It gives rise to a fairly easy type of covariant structure. And so he simulated this, and what he demonstrated is he could show some improvement in realized genetic gain over augmented designs. So this is an area of work that I think we need to spend some time on. And I've thought about it, but just haven't spent any time on it. So if anybody's there that's interested in it, I'd be glad to talk to you more about this. There is software available, bigger software. I've never used it. I think it's out of Australia, and that might be worthwhile to look at if you'd like to do these kinds of things. So here's the schematic that came out of Colas's paper, and you might look at it. He had three different assumptions about the varieties or the entries. He assumed that if lines are fixed or if they're random and with a certain size genetic variance, or if C is random with another size genetic variance. And so you can see how the pattern of these designs change. He used an algorithmic optimization routine to identify these designs, and that's all in Digger, I believe. So now the last kind of designs I talk about are unreplicated designs. And people have been doing these kinds of things for probably well over 100 years, maybe a lot longer than that. In terms of, you think about how people been selecting plants for thousands of years, well, they probably weren't using much of a design. So about 100 years ago, at least one of the first designs that I think in the literature that I've seen was a traditional design where you have a block of plots. In this example, I have 9 plots in a block, and you plant a check in this group of 9. Okay, and I got a second group of 9, I put the check right in the middle. And then to evaluate these, what you could do is you could say, okay, I'm going to choose the entry in each of these blocks that is the largest above the check that I can get. So I could go into each block and choose an entry. Similar types of things have been suggested over the years to put more than one plot in, or block check in. And of course, this is similar to an augmented design. And so it's a similar type of thing. Mass selection is basically a certain type of design. Charlie Gardner, a man that taught me a lot of stuff, who's since retired, a famous maize breeder from Nebraska, did a lot of work on mass selection. And basically the idea of mass selection is what other people call grid plot design. You take your whole field and plant all your entries out all with a single plot, and then you break it up into grids, and then you go within a grid and choose the one that's the best. Okay, and this is maybe better than just going and choosing one in the whole field, because you're trying to remove the variation within the field. But all these unreplicated types of designs give rise to, well, how are you actually statistically going to test these things, and how do you go about significance testing? And as long as you have some repeated checks somewhere, then you could fit a spatial model. And the idea of a spatial model is, say, here's the model for a simple randomized complete block design. We can write it generally with matrices, where these are the fixed treatment effects, say, and these are the random block effects, and these are the residual effects. And we make the assumption that we have certain block effects that have a certain simple structure to them, but that also we have an error structure that comes out through this correlation matrix R. And so we capture the spatial correlation between the plots within a field with this covariance matrix R. Okay, and so, well, we could estimate the slope coefficient or the effect of the treatments with this generalized least squares estimator. And if we're going to choose a design, what we could do is we could choose X, which is really the location of the different entries, to minimize the inverse of this particular matrix. Now, of course, that's a matrix. How do you minimize a matrix? Well, you'd have to use the determinant or something. But these are the kind of ideas that people have used with unreplicated designs is to try to use spatial ideas to generate kind of use an algorithmic method of selecting designs. The paper by Cibolli was a Nebraska student a number of years ago and did some simulations and found that with unreplicated designs with replicated checks, that a Knight's move type of design, notice that the check goes down 1 and over 2. That's called Knight's move, down 1 and over 2. That the Knight's move was pretty good with 20% of the plots, okay, given certain types of field variation. And she also found that, well, we could have a kind of a bigger type of thing like a Knight's move. Here we go down 3 and over 1, down 3 and over 1, okay? And that with 10% of the plots, this is a kind of a design that she found to be optimal. But there's a lot of need for work in this area also, is how do we get designs that are robust to different types of spatial variability, so we don't need to know what kind of spatial variability we have. I don't know. Is that possible? Well, maybe, but I don't know. Final thing I'd like to talk about are multi-environment testing. Seems like to me most of you people are doing studies throughout several environments, and then we need to talk a little bit about this. I'd just say that there's not a lot of issues to deal with other than how you analyze the genotype by environment interaction. But in terms of the design, any type of design can be used. You just use a different design or use the design at each location and make sure you randomize it at each location. Mike Talbot a number of years ago published a paper that said, well, in most field crops, that if you're going to use multi-environment testing, that for most field crops, you probably need 12 sites to really get a good idea of how well your entry is doing in farmers' environments. And this kind of harks back to a paper written by Sprague and Fetter years ago, where they were talking about similar types of issues and how many replicates do you need at a site and how many sites do you need in order to maximize genetic improvement of single cross corn hybrids. And so these are nice papers if you're interested in designs for multi-environment trials. But the interest really has not been so much in the design of these kinds of trials. It's been more of the analysis and how do you analyze genotype by environment interaction. Okay, conclusion. I did this in 45 minutes, well, that's great. Hopefully you have time for questions. My conclusions here would be that probably the most important thing about all of this, if you had to remember one thing, is you need to block. The most important thing is to try to block your field in such a way to remove that field variation. And however you do it, well, maybe that's not as important as doing it. And, you know, unfortunately, if you look at a lot of the crop science literature now, there's an awful lot of people that are doing these big experiments and they're not adequately blocked. That, you know, there are sometimes hundreds of entries in a field without any blocking at all, without any consideration to dealing with that variance. So I'd say that's the most important things. Also say that there are many different kinds of designs. Hopefully I give you some ideas that if you have a problem, that you can see how to at least kind of see the literature and know where to go. And finally, the best choice of a design is kind of practical trade-off between cost, simplicity, and validity. I do have some references here you can look at. I didn't refer to all of them, but I thought that'd be a good group of references if you want to pursue this further. So, Captain, that's all I have to say. I mean, I'll answer questions if anybody has some.
You might want to just flash through your references real quick so it's on the
Is that what you mean?
yeah, just so people, if they watch it later, can see where they're at. Looks like people are typing.
I got a couple comments here. Scott Haley says, can you comment on the choice of fixed versus random for entries? I have seen some discussion of this in the Australian literature. This is kind of a philosophical issue. I mean, for me, it kind of depends on your problem. Even before you go to the field, you need to know what population do you want to make a statement about. Are you really talking about a random sample of entries from some bigger population. I suppose if you say I take a cross of inbred line 1 versus 2 and you generate all the progeny, I suppose technically those progeny can be considered a random sample from all the possible progeny you could produce from that cross. However, if you're trying to make a statement about all possible progeny you could produce from that cross. However, if you're more interested in making selections, you're not interested in, you don't give a hoot about making a statement about a population. You want to know if that one entry right there is better than the other one. Is it better than the check? In that case, if you're more interested in selection, maybe at the latter end of a breeding program, I would think that considering entries to be fixed would be preferred as opposed to random. Usually at the beginning of a program, you have many hundreds, maybe thousands of entries. In that situation, often people consider them to be random. So I'd say the most important thing is to know what your population is. What do you want to make a statement about first? If it's more limited, I want to just evaluate those 3 entries, those ones for release, then I'd say they're fixed. If you're at the earlier stage, you have a population, want to make a statement about an entire population of all possible progeny that could come from a cross, I'd consider random. So whether that makes sense or not, I mean, to me, just go back to what is the population you want to make a statement about. I have another question here. Let's see. I have just started my way to become a breeder, so I would like to ask, what is the minimum size of yield trial? And it depends on what? Well, what are your objectives first, I would say. Let me make it more simply. Let me apply it to say something like a clinical trial. I have 2 drugs, drug A and B. And the question is, people always incessantly say, how many subjects do I need to have a certain belief in my results? And what the FDA mandates for comparing drugs is that you show that you have enough people on each treatment to have an 80% chance of showing a difference of a certain size. And you have to lay this all out in your protocol before the FDA will approve the trial. So it would be a similar thing in plant breeding. Depends on what your objective is first. If you have just 2 varieties that you want to consider for release, and you're going to consider one or the other depending on whether one is significantly higher than the other, then you need to make sure you have enough plots and enough a good sampling of environments where they're likely to be grown, so as you can have a high level of confidence that you're going to pick up differences between treatments if the differences between entries is really true. I hope I answered your question. Another question here. Do you recommend including multiple checks of, say, the same susceptible variety per block if you have a randomized complete block design with 2 or more blocks and 400 entries? Well, it's hard to say. I mean, depends on what your objective is. You say susceptible variety. If you're talking about disease testing, Lord, I don't know. I mean, you'd have to decide what the goal is. But if you've got 400 entries per block, I don't know. You better have at least a few entries in there. But it depends on the plot size. 400 entries per block is too large. I'd worry more about getting the block size smaller than that. So I don't know. At least a couple for Fetter's augmented designs. He generally assumed you have to have at least 2 checks, and often more than that. Okay, another question. Why is randomized complete block design inefficient? And what are your thoughts on a split plot? Well, the complete block, all I'm really assuming here is that if you have a lot of entries, say you have 100 entries, if they're maze plots you're talking about, that's a pretty big piece of ground. And it's unlikely you're going to be able to have a uniform piece of ground with 100 maze plots all together. And so I'm just making that assumption from the very beginning, is that you have a lot of entries to look at. The block size is going to be large. It's going to be, complete block size is going to be too large. And because of that, if you have too big of a block, or complete block, there's a lot of variation within the block, and that goes out into your air turn. Well, you don't want that noise in the air. You want to remove the noise from the air. So if you have incomplete blocks, then that makes it less. The split plot design, I would say for plant breeding, the split plot designs are generally not very useful because you really only have one treatment factor in most plant breeding experiments, the treatment factor is entry. Okay, but in some situations you do have several factors, and given the situation, then maybe a split plot would be called for. What I tell the students in the advanced design class here is that avoid split plots if you can, because they're more complex, and often you lose more precision on the main plot factor than what you gain on the split plot factor. So there's a real cost with using split plots, but sometimes you can't do anything else. And that's when I think you have to use them. For example, you've got irrigation, you got fields are irrigated, and the field is the whole plot factor, and you put your entries within each irrigation level. Sometimes you can't do anything else but assume that the whole plot is split plot design is appropriate. And of course, if that's the case, then so be it. Then you have to use a split plot. But I generally don't recommend them if you can avoid them. Just because you think students say, "Oh, I get more precision in the evaluating split plot factors as compared to the whole plot factors. Therefore, I want to use a split plot." But the problem is you lose so much on the whole plot to gain only a slight amount of increased precision on the split plot. So if you're willing to make that sacrifice, I suppose that's fine.
Do we have any other questions for Dr. Eskridge?
Well, okay, thank you. I hope that was helpful. And just send me an email if you're interested in any of this. I'd be glad to talk to you about it. Well, thanks a lot.
Yeah, thank you for starting the seminar series off well. We'll be continuing these webinars every other week throughout the semester. So our next coming...
Just, if we mute that, that'll...
As I was saying, we'll have these seminars every other Wednesday throughout the semester. So our next one will be on October 17th at 3 p.m. Central. We'll have Dr. Paul Gepps from UC Davis, and his webinar title is going to be In Situ Farmer Management of Genetic Diversity as a Complement to Ex Situ Conservation. So these recordings will be available on the Plant Breeding Training Network website, as I think will be PDFs of the slides. So thanks everyone for coming, and thank you again, Dr. Eskridge, for the great presentation.